providing an update on the current status of our biological control program for introduced Phragmites in Canada. I'm going to be co-presenting this with Ian Jones, and we're presenting also on behalf of colleagues Sandy Smith from University of Toronto and Rob Boucher from Agriculture and Agri-Food Canada. The invasive species we're dealing with here is, of course, introduced Phragmites, so Phragmites australis subspecies australis, and this has been named as one of the worst weeds in North America. This is a wetland species that is capable of forming dense near monocultures, as we can see here, that then go on to have a wide range of negative ecological and socioeconomic impacts. To supplement the variety of tools that we already have to manage introduced Phragmites, our team has been working on providing what we like to think of as another tool in the toolbox for Phragmites in the form of biological control. Biological control, or biocontrol, uses living organisms to gradually suppress a pest like Phragmites at large scale over the long term. And there are many advantages to biocontrol as a method. We are reuniting a weed with its natural enemies that it might have left behind, thereby reestablishing an ecological balance that can help keep that weed in check. Because we're only using agents that are highly specific to their host organisms, this technique is very safe with low off-target environmental impacts. And finally, because these are living organisms that reproduce and disperse on their own, this method is also highly cost-effective, including at very large scales and over the long term. Biocontrol agents that we have for introduced Phragmites are two European moths with stem-boring larvae. So you can see here Arcanera neurica and Lanisa geminipuncta as larvae and as moths. The larvae are the life stage that do the actual damage to the plant. As they develop, they will mine through 3 to 4 shoots of Phragmites, leaving behind boreholes as they enter and exit the stems and killing off Phragmites tissue above that feeding point. This feeding damage will typically kill young stems outright, whereas older stems will be stunted. They'll survive, but they're not able to grow past that feeding damage. And so as a result of this feeding, we see Phragmites reduced in dominance and competitiveness, leaving space for other desirable biodiversity to reassert itself. Biocontrol for Phragmites has been in development since 1998. At this time, an international team worked to identify the agents that we are currently using and conducted extensive host range testing to ensure that they are specific to the target weed. So they are only able to feed and complete their life cycle on the target plant. Once this was demonstrated, this led to the approval to release in Canada. The Canadian release permit was approved by the Canadian Food Inspection Agency in 2019. With that permit in place, this then led way to the first Canadian releases in Ontario. The first years of the project, from 2019 to 2023, were focusing on developing a set of core operational protocols for how to rear large numbers of the biocontrol insects, how to release them onto the landscape effectively and efficiently, and how to monitor their dispersal, reproduction, impact, and so on. And so during that time, we emerged with a set of these core operational protocols. So with those best practices in hand, the project is now moving on to a third phase of essentially scaling up releases across Southern Ontario. Key to this strategy is the use of what we would call a nurse site. These are locations that have the agents present at them with very large, robust populations that maybe have developed over a few years. And these nurse sites will help facilitate spread to other parts of the landscape. This can happen as the insects themselves will disperse locally, flying to nearby Phragmites. And we're also looking at methods of assisted redistribution whereby we can go in, harvest agents from these large populations, and then actively move them to sites of our specific choosing. From 2019 to 2023, we've released approximately 21,000 insects across 30 sites in Ontario. As you can see, we've made some effort to distribute these across different geographic regions to get as much coverage as possible. We collect monitoring data on damage that the insects do in terms of feeding damage, number of damaged stems per square meter of ground. We see that as a summary here across our 30 release sites. We collect that data in the very first year when we release, and then also we will go back in subsequent years to track how that population is doing. We can pull out a few general takeaways from this monitoring data at this point. The first is that the releases work very well. So Using our best practices, we release the insects, we go back a couple of months later, and we're able to see initial feeding damage at 92% of those release sites. So to give you an idea of what this might look like, the PVC marker here is denoting one of these release points where eggs or larvae were placed. Each of these blue flags is a Phragmites stem that has been fed upon and damaged by the agents. So we're able to quantify that, and we're seeing here a couple months after release, damage and dispersal away from that release point, which is what we want to be seeing at this point. We can also look to the subsequent years. So these would be years where we're not actively releasing agents, we're going back to monitor how they're doing on their own. And what we see here is that, encouragingly, damage persists. So all sites that had damage immediately following release continue to have damage into the second year, and now we have a third year of monitoring data as well. And we said that damage is generally consistent or increasing. So similarly, we're able to go back into these sites, we can quantify the number of damaged stems that we're finding. But what's really exciting about year 2 onwards is that this is damage from the offspring of the insects that we released. So when we see this damage, we know that they are able to reproduce and overwinter on the site. And finally, we're also seeing that agents are able to disperse locally within release sites. We usually release at only a part of a site, but we are able to find damage in other parts of those populations as well. So for example, this is a release site near Aurora where we released larvae and eggs around the perimeter of the patch in 2020 and in 2021. By the following year, we run monitoring plots through the center of the patch, and we find that the vast majority have damage there. So this is a part of the habitat that they have moved to on their own within a short period of time. So to wrap up this first part of the presentation, in those first few years of the Ontario program, we've seen the development of these core operational protocols. We've seen them put in place with very encouraging monitoring results from the first 3 years. So looking forward, the increasing focus is going to be on scaling up these releases across Southern Ontario and really trying to build an even more extensive network of nurse sites across this geographic range. So here, I'm going to hand things off to Ian to continue.
Thanks, Michael. With any kind of early stage biological control release program, all these releases and monitoring need to go hand in hand with a directed research program, some of which Michael has touched on, but it's to give ourselves and practitioners and land managers the best possible information to actually make a successful management tool out of this biological control agent. So for the rest of this talk, I'm going to focus on some of that research that we've been doing over the last few years. And broadly, that research falls under 2 questions. So how can we improve the success of insect releases? And in that case, I'm going to talk about an experiment we've done looking at the effects of hydrology on release success. And secondly, how can we maximize the number of insects that we actually have to release? And again, as Michael kind of alluded to, what we're looking to hopefully do is move away to some degree from a reliance on lab-reared insects and move towards a nurse site strategy whereby we can identify Phragmites infestations in the field where we know we have solid populations of these agents, and then move those agents directly from those nurse sites into other Phragmites infestations. So next slide, Michael. Yeah, so that first question I mentioned is kind of how can we improve the success of our releases? And we're looking in this experiment at the effects of hydrology. So the site that you're looking at there is at Lynn Shores Conservation Area. Along that kind of leading edge of the Phragmites, there's about 30 centimeters of standing water. And then along the other side of that same linear patch, there are much drier conditions. So we conducted 20 Arcanara and Eurica releases along 10 along the wet side and 10 along the dry side of that patch. And in each case, we're doing 5 egg releases and 5 larval releases. Next slide, Michael. So this is what those releases look like. You can see those wet releases on the right-hand side. So any kind of first instar larvae dropping out of those egg containers or more mature larvae eating their way out of those transferred stems are going to have to traverse some of that standing water before they reach the growing Phragmites that they want to infest. So the question we're asking here is, does that standing water represent an obstacle to the success of these release methods? So the dry releases that you can see on the left-hand side there, they're much more kind of representative of the kinds of releases we've tended to conduct so far in the program. So about 6 weeks after these releases, we monitored those release points in the same ways that Michael described, looking at the numbers of damaged stems surrounding each of those release points. So next slide, Michael. So here's what the data looks like there. You've got the number of damaged stems along the y-axis, and then you have the results for both egg and larval releases in dry and wet conditions. So the first thing you'll probably notice is that those larval releases tend to perform much better. We get more damaged stems. And this is something we've grown to expect. Those larval releases where we inoculate stems are the most effective release form, but they're also labor intensive. So there's a trade-off there, and there's an advantage to both of those release methods. The other thing you'll notice is that the pattern of success between the dry and the wet conditions is more or less identical. If anything, we see somewhat better performance of these releases in the wet sites. So that's incredible news for the biological control program at this point. It tells us that we can conduct these releases in a range of hydrological conditions and not be too concerned about any loss of efficacy in doing so. So this opens up a wide range of new potential release sites for us. All right, next slide, Michael. So the other question we're addressing is how can we improve or increase the number of insects that we have at our disposal? And again, one of the things to do that is to move up to a nurse site strategy whereby we're no longer so reliant on lab-reared insects, which are time consuming and labor intensive and can be expensive to produce. If we can just transfer insects from one nurse site to new Phragmites infestations, we can save a lot of time and be more efficient at spreading these insects through the landscape. But the idea of collecting insects from insect eggs, for example, from a huge field of Phragmites is quite daunting. But that probably is the best way to collect and transfer these insects, is to collect Phragmites stems late in the season in the hope that they contain our agent eggs, and then move those stems into new sites. But in order to kind of determine how effectively we can do that, in the past summer, we've been visiting several of our nurse sites, collecting stems and bringing them back to the lab to dissect them to see how many eggs we've harvested. So to give you one example, from one of our nurse sites, we collected 116 stems. We dissected them in the lab and found 367 agent eggs. So we were really happy with that, roughly 3 eggs per stem. And it was a fairly quick way to amass those numbers of eggs that can be useful for future releases. The data on the bottom on the left shows the positions on the Phragmites stems where those eggs were found. So the vast majority of eggs seem to be laid low down on the stem in the lowest meter or so. So what that means is that when we come to transfer these stems, we don't have to carry around the whole cumbersome 5-meter tall stem. We can cut it down to much more manageable size and then transfer them in setups like you can see on the right-hand side there. So we have a kind of vertical grid of stems whereby when the new Phragmites grows up in the spring, it will grow in and among those stems and make it very easy for any emerging larvae to infest the growing Phragmites. And so for the first time this year, we have 18 of those releases set up at sites at Ontario Tech University. And we're excited to be monitoring those in the spring and early summer to see how well those releases go. So next slide, Michael. So yeah, to summarize, since 2019, when these releases began, we've released about 21,000 of these insects across southern Ontario, across 30 sites. Early monitoring results have been really, really encouraging. We're seeing successful reproduction and overwintering at the vast majority of sites. And at the sites where we have multiple years of monitoring, we tend to see increased populations, or at least increased damage caused by these insects as the years go by. So we're really happy that we've developed effective release methods for both eggs and larvae, and happy to see more recently that those methods are successful across this range of hydrological conditions. So that opens up a huge range of new potential release sites for us. We've been surprised and very pleased with the degree to which we've been able to harvest agent eggs from Phragmites nurse sites. And so those stem transfer releases will be happening for the first time in 2024. So in the short and medium term, the goal for the project is to scale up the release program and to continue to amass this network of nurse sites. And depending on how these different transfer releases go, that might be a way to hugely speed up and accelerate the process of doing that. So next slide, Michael. Yeah, so now just time to say thank you to all of our supporters and collaborators. Many of them are here today. And I'll kind of just leave this here for a few minutes before I open up for questions. Hopefully we have still a little bit of time.
Great, thank you so much, Michael and Ian, for that great presentation. So we have tons of questions in the question and answer box for you. So I'll start with the first one. Do you think it is possible for these biocontrol agents to mutate and start feeding on native plant species over long term?
So one of the main steps that happens in these processes is based on safety in that permitting, so ensuring that initial host specificity. Beyond that period, in terms of, so we know that right now they're just eating introduced Phragmites. Beyond that, that kind of transfer is not a concern and not something that we would expect from the biocontrol agents, in that process is no more likely to happen to the biocontrol species than to any other native species that might be present in the environment as well. We also know that there are a variety of factors that will naturally constrain their populations over time, including competition with themselves, availability of food resources, novel predators that'll keep their populations in check, and so on. So no, that is not a concern.
Great, thank you, Michael. Next question. I'm wondering if there's any risk of the moths causing harm to native species.
Yeah, so I think as Michael mentioned, the host range testing for these potential biological control agents is extremely rigorous at this point. So this is kind of, in the case of these agents, an 11-year process. So it's the centrifugal phylogenetic method, whereby the most closely related plants that will share the introduced range are tested first. And they're tested to see whether the insects will feed on them, will lay their eggs on them, can complete their life cycle on them. And then this is kind of expanded out to a much larger number of plants that are maybe more distantly related, or might be of economic or cultural significance. And it's only after we're completely satisfied that they're very highly specific to the target weed that they would ever even be considered as biological control agents. So in terms of modern weed biological control, the risk of non-target effects is very, very low.
Great. Next question, were some sites not measured for damage in year 2 and 3, or did most have no damage recorded?
So thinking about that grid, that table of our monitoring results, the fact that we're not seeing some for years 2 and 3 is just a consequence of how old the sites are. So we add new sites every single year, and we continue monitoring on an annual basis. So after another year, we'll be filling in more sites with year 2 data and so far. So what you saw there was just the old sites that we have so far, with more and more sites coming down the pipe every year. So that'll be a database we continue to grow as the project develops.
Okay, we have a couple more questions. There's quite a bit more, lots of interest. So maybe after a couple more, 1 or 2 more, if you could go answer in the chat box, Michael and Ian, if you're okay with that. So let's, does anyone track the behavior of their distribution, survival, and effect on native species since 1998?
So the program was started in 1998, but that was the very early stages, including doing the foreign exploration to find potential agents, and then all of the host range testing and the safety testing that goes into it. The insects have only been released in Ontario since 2019. And yeah, so at the moment we're monitoring very closely where they go, but also developing methods to kind of monitor on more of a landscape scale through things like the development of pheromone traps, and also the use of drone imagery to try and see if we can actually identify some of the damage that these insects are causing from drone images. So yeah, there is very much a kind of a process by which we track their movement and their activity over the early years of the releases.
And I would add one thing we look at as well, and another example of the level of specificity that we have with these agents is that we are selectively working at sites where we have our introduced subspecies of Phragmites in close proximity to our native subspecies of Phragmites, which is very similar in many ways, but is of conservation value. And we are seeing specificity to that introduced Phragmites. So they're not even spreading to a very closely related, morphologically similar plant as well, which just further reinforces from field data what was seen in that host specificity testing that comes before that permitting process approves them for release. And that's something we'll continue to monitor as time goes on as well.
Great. Thank you so much, Michael and Ian.